Good afternoon, everyone. It's my pleasure to be speaking at this event. I would like to thank Thermo Fisher for inviting me to share my work with you. During this pandemic, virtual education events such as this one are very valuable and help make quarantine a little bit more interesting. Today, I'm going to talk about molecular profiling of hematologic malignancies by next-generation sequencing. This is the disclaimer. The prevalence of hematologic disorders in U.S. in 2019. This data is from Leukemia Lymphoma Society. Every 3 minutes, one person in the U.S. is diagnosed with a blood cancer. About 176,000 people in the U.S. are expected to be diagnosed with leukemia, lymphoma, or myeloma. New cases of leukemia, lymphoma, and myeloma are expected to account for 10% of the estimated 1.7 million new cancer cases diagnosed in the U.S. About 61,000 people were diagnosed with leukemia in 2019, and 14,000 people were diagnosed with MDS. About 9,600 cases of MPN were diagnosed per year. Let's look at the guidelines from NCCN. The first guideline I'm going to talk about is the acute myeloid leukemia, we call it AML. It is recommended that cytogenetics analysis, including karyotype and FISH, to be done. For molecular analysis, including c-KIT, FLT3, IDT, and TKD, NPM1, CBPA, IDH1 and 2, TP53, and other mutations to be tested for the patients. It is recommended the maximal turnaround time for the assay is about 3 to 5 days for immediately actionable mutations, including those genes. In particular, for patients with FLT3 positive mutation, the patient has to be on the treatment in 8 days. The next guideline is for MDS. On the left side, highlighted area are the genes with the prognostic information for MDS. The third guideline I'm going to talk about is for myeloproliferative neoplasm, which is MPN. The molecular testing on the blood for JAK2 V617F mutation has to be tested first. If it's negative, test for CALR and MPL mutations for patients with ET or MF, and the JAK2 exon 12 mutations for patients with PV, and the molecular testing using multi-gene NGS panel, including those genes, are recommended. There's also guidelines from CAP and ASH for AML genetic testing recommendations. It is also recommended, very similar to the NCCN guideline, that include karyotype and FISH, and also molecular testing for all or most of the cases with those mutations I just mentioned, which is very similar to the NCCN guideline. For selected cases, the kit for core binding factor leukemia, and also for APL, we have to do the PMR-RARA test. Next, I'm going to talk about genetic abnormalities in AML. This is a very busy figure. If you look at on the left side, there are many genes. Their mutations commonly seen in AML patients. You can see all the percentage represent the incidence in the AML. On the right side of the slide, there are many translocations associated with AML patients. Some of the mutations coexisting with the translocations that will change the prognostic information or the treatment. The treatment associated with myeloid biomarker testing, let's look at the disease for AML. The biomarkers include FLT3, IDH1, and 2. The assay used are PCR and NGS, and also the drug on the right side of the column. For CML and APL, there are BCR-ABL1 and PM-ARARA translocations. The assays include the PCR FISH karyotype NGS, and also there's drugs on the right side. The APL use the ATRA, for example. For the PV, the JAK2 mutation analysis is recommended to use PCR or NGS method, and use the drug is also listed here. For so many different biomarkers and tests in these patients, tests usually performed sequentially, and it takes a long time to give results. It uses many samples instead of one. Therefore, NGS is the solution to reduce number of assays, turnaround time, and the sample size. If you think about the figure I showed in the previous slide for AML, there are so many mutations and the translocations. If you do it sequentially, it will take a really long time to get results. The clinical utility of the multiple biomarker testing by NGS, the detection of multiple mutation fusions in target genes in a panel makes huge impact on screening, diagnosis, prognosis, monitoring minimal residual disease, and predicting each individual's response to treatment. Next, I'm going to show you two examples that NGS has helped with the diagnosis of the patients. First one, the indication is 70-year-old male with ALL, chemo-induced pancytopenia. The tests ordered include FISH, BCR-ABL1 quantitative assay, karyotype, and NGS. The results showed for FISH is positive for BCR-ABL1 fusion. However, the BCR-ABL1 quantitative assay did not detect either P210 or P190. For karyotype, it is QNS without results because insufficient sample. Let's look at the BCR-ABL1 transcripts. The typical BCR-ABL1 fusion transcripts include P190 and P210. So on the left side, the blue color shows the BCR exons, and on the right side is the yellow color highlighted for the ABL1 exons. If it's E1A2, stands for P190. So for P210, there's two forms. One is E13A2. That means the exon from BCR E13 fused with the ABL1A2 exon. And for E14A2, it's also P210. For atypical BCR-ABL fusion transcripts include the following: E1A3, E13A3, E6A2. For E19A2, that is the rare transcript for P230. This test is provided by a limited of the laboratories. Most of the lab, they have P190 and the P210. So for this particular patient, we run the NGS, and NGS could detect BCR-ABL1 transcripts for common and uncommon forms. On the top is E13A2 for P210, which is a common form detected from a different patient. on the lower part of this slide, for this particular patient, we detect E13A3, which is a rare transcript. Clinical case 2. The indication is a 53-year-old man with AML history. Test ordered include karyotype, FISH AML panel, and NGS. The FISH show the results negative for AML panel, including RUNX1, RUNX1T1. The karyotype showed in the slide, it is loss of Y, added 1Q, deleted 8Q, added 21Q in 3 out of 20 metaphases. So what is the NGS results? The NGS result showed that there is a RUNX1, RUNXT1 translocation. Next, we went back to re-examine the karyotype. Then we figured out it is actually a 3-way translocation. So the part of the 1Q goes to the 21Q, the 21Q goes to 8Q, and 8Q goes to the 1Q. Then we re-examine the FISH results. We analyzed the additional 300 cells. 3 out of 500 cells showed 1 fusion, which is a yellow signal in the interface cell, 2 green and 2 red signals. It is confirmed a 3-way translocation involves RUNX1, RUNX1T1 translocation. Take-home messages for these 2 cases. NGS for hematopoietic neoplasms may be performed on peripheral blood or marrow aspirate, may detect mutations, rearrangements, replacing some FISH and PCR-based analysis. Turnaround time equal to or more rapid than conventional assays. Our turnaround time is about 3 to 5 days. FISH still required, desirable for monosomies, large deletions, for example, chromosome 5 or 7. More rapid detection of PM-ARARA or other rearrangements. For case 1, NGS identified a rare transcript for BCR-ABL1, which is E13A3 fusion. For case 2, NGS detected a fusion that was missed by FISH or karyotype.